Hello Ji, Namaskar. Hi everyone. How are you all? Well, I'm your very own Dr. Sachin Kapur. And majority of you already know me, and those who do not know me, let me tell you that I have an MPhil and PhD degree, and I have been teaching students for the last 25 years. In these 25 years, I have got an opportunity to teach excellent students who are well settled in their profession, medical profession, not only in India but abroad also. So I wish the same for you people also. Well, you can follow me at various social media. You can follow me on my Instagram, and this is the WhatsApp number. This Telegram group is very important. You must join this Telegram group for any kind of information which is related to your medical studies, whether it is related to syllabus or you need any kind of study material. So please feel free to contact me. Another important thing, this app, the name of the app is Dr. Sachin Kapur itself. This caters to your needs related to NEET PG as well as NEET UG. This is completely free of cost, and you can install this app from Google Play Store. You will get all the material which is related to your anatomy, physiology and biochemistry. As you know that the syllabus which Indian universities are following these days for medical studies is completely competency based. That's the latest MBBS curriculum. The book which you people have to follow is Guyton and Hall Textbook of Medical Physiology, South Asian edition. Please remember, this South Asian edition has been designed keeping in mind the needs of students as per the syllabus of Indian universities, the competency based syllabus. Now, what we are going to discuss in this session is the MBBS first year physiology syllabus will be highlighting the subtopics of hematology. What all you need to study in physiology: general physiology, hematology, nerve and muscle physiology, gastrointestinal physiology, cardiovascular physiology, respiratory physiology, then renal physiology, the structure and functioning of kidneys, endocrine physiology, various endocrine glands, reproductive physiology, neurophysiology and integrated physiology. In this session, I'll be discussing in detail about hematology subtopics. You know what is hematology? The study of blood. This itself is a specialized branch of medical sciences. Now, listen carefully. What all we need to study in this topic hematology? What are the different subtopics? The first subtopic is blood composition. So we need to know the composition of blood, what is plasma, what is serum, and what are the functions of blood. Broadly, blood. What is the volume of blood in a normal, healthy adult individual? For males, it is 5 to 6 liters. For females, it is 4 to 5 liters. On an average, we say that human body has 5 liters of blood. Broadly, the composition of blood is, it has plasma, that is 55 to 60%, and then formed elements. Now, when I say formed elements, that includes red blood corpuscles, that is RBCs or the erythrocytes, WBCs or the white blood cells or leukocytes, and platelets or the thrombocytes. So 55 to 60% is what is plasma, and close to 40 to 45% is what is the formed elements, right? So in this subtopic, you should know details about plasma. What is the further composition of plasma? We know that plasma consists of water, right? In addition to water, there are other substances. Can you guess what are other substances? The other substances can be organic, these can be inorganic. 91 to 92% is water and roughly 8 to 9% is other substances. These other substances can be organic or these can be inorganic also. Now, when I say organic, it includes proteins as well as non-proteins. Can you think of different proteins present in plasma? Serum albumin, serum globulin, fibrinogen, prothrombin. So we'll be discussing the functions of all these proteins. Then there are other substances also: urea, uric acid, amino acids, glucose, lipids. There are lot many things which are present in plasma. So we have to discuss the detailed composition of plasma. Now, what is serum? You know what is serum? When blood clots, then after a few minutes, that clot retracts. When the clot retracts, a fluid oozes out of that clot, and that is what is called as serum. Serum doesn't include the clotting proteins. So we're discussing about serum in detail. Then functions of blood. There are different functions of blood. Blood is responsible for transportation of the substances, transportation of gases, oxygen, carbon dioxide, transportation of hormones, nutrients, metabolic wastes. It is distributing the body heat also. Then immunological functions are also there. So all those functions will be discussed in detail. When it comes to plasma proteins, we will be discussing the origin of plasma proteins. That is one subtopic. Another subtopic is the different forms of plasma proteins. We'll be talking about serum albumin, globulin, alpha, beta, gamma globulins. Gamma globulins are antibodies. What are the functions of plasma proteins? Fibrinogen and prothrombin are involved in blood clotting. Relation of diet to plasma proteins and variations in plasma protein concentration. So these are the subtopics of your competency based syllabus from plasma proteins. You should be thorough with your syllabus. It should be at your fingertips. Next topic in hematology is about hemoglobin. Hemoglobin, which is red colored iron containing oxygen transporting protein, which has 2 alpha chains, 2 beta chains. There are different forms of hemoglobin, right? There is adult hemoglobin, fetal hemoglobin, sickle cell hemoglobin. So the different varieties of hemoglobin we need to discuss. What happens when hemoglobin breaks down into heme and globin? Globin is the protein part, right? Reutilized. And heme is the iron containing part. We'll discuss how bilirubin and biliverdin are formed. What are the functions of hemoglobin? Hemoglobin not only carries oxygen, it carries carbon dioxide also. We'll be talking about oxyhemoglobin, deoxyhemoglobin, carbaminohemoglobin, carboxyhemoglobin. Carbon monoxide can also combine with hemoglobin, right? How hemoglobin is synthesized and the detailed structure of hemoglobin. So all these subtopics we'll be discussing in hemoglobin. Next topic is about erythropoiesis. Erythropoiesis means the formation of RBCs. We know our bones have bone marrow. There is red bone marrow, yellow bone marrow. Red bone marrow is the active bone marrow. So hemopoiesis, that is formation of the blood, occurs in the bone marrow. Erythropoiesis is the formation of the RBCs. There are different stages in the formation of RBCs: hemocytoblast, proerythroblast, early normoblast, intermediate normoblast, late normoblast, then reticulocyte, erythrocyte. We'll be talking about the different metal ions which are required, vitamins which are required for the production of the RBCs. The structure of RBCs. We know that mammalian RBCs are well suited for performing a particular function. Their shape allows them to squeeze through the narrow capillaries, and they have enough surface area which allows the diffusion of the gases across the erythrocyte membrane. Majority of the cell organelles are absent. There is no mitochondria, no Golgi, no centrioles. So absence of majority of the organelles provides enough space for carrying more and more hemoglobin. Metabolism is anaerobic. Mitochondria are absent. What would happen if mitochondria are present? Mitochondria will themselves utilize oxygen. So then how will be the RBCs capable of transporting oxygen, right? We'll be discussing about variations in size, shape and structure of RBCs, increase in RBC count, decrease in RBC count, then the different theories for hemopoiesis, and erythropoiesis in detail, and regulation of erythropoiesis, how hypoxia stimulates the production of more RBCs, what is the role of renal erythropoietic factor. So all these subtopics are part of your competency based syllabus for your MBBS. Anemia, in detail we have to discuss about anemia. Anemia is basically the decrease in the number of RBCs, which ultimately result in decrease in the amount of hemoglobin also. There are different ways of classifying anemias: etiological or Whitby's classification, morphological or Wintrobe's classification. What is sickle cell anemia? What is pernicious anemia? Megaloblastic anemia? So even thalassemia, all these things will be discussing. Thalassemia, sickle cell anemia, erythroblastosis fetalis, or the hemolytic disease of the newborn, when the Rh factor of the mother and the fetus doesn't match. If an Rh negative female is carrying an Rh positive fetus in her uterus, then what can be the complications? What are the type of antibodies which are produced, and what are the problems which they can create? This will be discussed in detail in erythroblastosis fetalis. G6PD deficiency, that is glucose 6-phosphate dehydrogenase enzyme deficiency. We also call it favism. I'll be discussing these in detail. Do you know that the heterozygotes for sickle cell anemia, they have very less chances of getting affected with malaria, right? And even those who suffer with glucose 6-phosphate dehydrogenase enzyme deficiency, they do not suffer with malaria generally, because malarial parasite Plasmodium is not able to complete its life cycle successfully in such RBCs where glucose 6-phosphate dehydrogenase enzyme is absent. Then comes jaundice. The definition of jaundice: the chemistry of bilirubin formation, fate of bilirubin, what happens when the bilirubin is broken down, what are the different types of jaundice, and what is phototherapy. We'll be discussing about jaundice or the icterus in the newborn baby also, because the RBC count of the newborn baby is very, very high. So once a baby is born, majority of the RBCs, they undergo massive erythroclasia. RBCs are destroyed, large amount of bilirubin and biliverdin is produced, resulting in jaundice in a newborn baby. Then leukopoiesis, leukocytes, that is the WBCs. So we'll be discussing how the granulocytes are formed and how agranulocytes are formed. We know WBCs are of two types: granulocytes, which have enzyme-rich specific granules like neutrophils, eosinophils, and basophils. Then agranulocytes, which do not have enzyme-rich specific granules, includes lymphocytes and monocytes. So all those types of WBCs will be discussed in detail: their structure, their function, variations, regulation of granulopoiesis, and the phagocytic mechanism. How neutrophils and monocytes are capable of performing phagocytosis. What is diapedesis? All these subtopics will be discussed in leukopoiesis. Then the third type of the formed elements are the platelets, which are also called as the thrombocytes. So we'll be discussing about thrombopoiesis. Platelets are not cells, they are cell fragments which develop from megakaryocyte of the bone marrow. We'll be talking about structure of platelets, then thrombocytopenia, count and variations, functions of platelets. Platelets are involved in blood clotting, right? Then another very interesting topic, that is hemostasis. Please do not confuse it with the homeostasis. Homeostasis is a maintenance of constant internal environmental conditions irrespective of the changes in external environment. That is a separate topic of general physiology. It is a hemostasis, prevention of the blood loss. Suppose you suffer with an injury, there is loss of blood. Then how the blood is going to clot? What are the different steps which are involved in hemostasis? There is a vascular spasm, there is formation of the platelet plug, then there is formation of the clot, then there is retraction of the clot. So we'll be discussing about all the clotting factors in detail. That is fibrinogen, prothrombin, tissue thromboplastin, calcium, labile factor, serum prothrombin conversion, accellerin, anti-hemophilic factor, and lot many other factors, right? Then the different types of hemophilias: hemophilia A, hemophilia B, hemophilia C. fibrinolytic system, the natural fibrinolytic system in the body, that why the clots which are formed inside the body and why and how they get dissolved, what is the role of plasminogen, anticoagulants. Your body has natural anticoagulants, and even there are certain artificial anticoagulants also which are added to the blood when we store the blood in the blood banks. So sodium oxalate, potassium oxalate, how do they function? Then EDTA, ethylene diamine tetraacetic acid. So all those will be discussed in anticoagulants. Hemorrhagic disorders, disorders which are related to blood. Blood groups, very interesting topic. You have been studying this topic even in your junior classes. We generally talk about ABO blood group system and RH factor blood group system. But do you know that as per the International Society for Blood Transfusion, there are more than 30 different blood group systems? There is MNS system, there is a Lewis system, there is a Duffy system, there is Kell system, and lot many others. So why only ABO and RH factor systems we generally study? Why these antigens are more important from transfusion point of view? We'll be discussing that. The different blood group systems, then the clinical importance of blood grouping, right? Blood banking, blood transfusion. In ABO system, we'll be talking about people who have A blood group, B blood group, O blood group, AB blood group, and even the Bombay phenotype. Then what is golden blood? All these subtopics will be discussed in blood groups. Then comes immunity. What are the different lymphoid tissues in the body? The primary lymphoid organs, secondary lymphoid organs. We'll be discussing about thymus. Then we'll be discussing about the lymph nodes and even spleen. Then what is lymph? The different types of immune system which our body has. We have innate immune system, acquired immune system. What are immunoglobulins or the antibodies? The types of antibodies: IgG, IgA, IgD, IgE, and IgM. We'll be discussing the structure of all these antibodies in detail. Then regulation of immune response and immunological tolerance. How your body is capable of knowing this thing, that what is self and what is non-self. How autoimmune disorders they happen, right? Then at last we have this portion which is part of your practical syllabus. You have to find out the platelet count, you have to estimate hemoglobin, RBCs, total leukocyte count, that is TLC, right? Differential leukocyte count, blood groups, then ESR, erythrocyte sedimentation rate, hematocrit value. Hematocrit is the percentage of blood volume occupied by RBCs. So these are the topics which will be discussed in your practical classes. So that was about hematology. Right? Through these short sessions, I am making you acquainted with your syllabus, which is very important. Once your syllabus is on your fingertips, you get a confidence. You know that what all topics you have to prepare strongly for your exams. Right? Once again, these are the different social media where you can follow me on my Instagram, on Telegram, and you can simply search about me in Google, Dr. Sachin Kapur. And please do not forget to press the like button, hit the bell icon, and subscribe my channel. And please do take out time to see the description of the video also. So I wish you all the best for a very bright and prosperous future. Stay blessed. God bless you. Namaskar.